All right, Mr. Sapora here, and this week we're going to be talking about DNA and its structure, what it's made of. This is going to involve a little bit of chemistry and some vocab, but don't freak out if you've never had any chemistry. The beauty of DNA is that it's simple and it repeats itself again and again and again, and that's what allows it to be a code. So once you learn kind of the four basic parts or so of this molecule, you'll be able to understand it when we get into next week's topic, which will be how does DNA actually create protein. If you remember from the first week when we just introduced DNA, we talked about how DNA is this long molecule. Hey, look, I found something. And there are sections of this molecule that carry information, right? Like paragraphs or chapters in a book. And those sections that carry information are called genes. And a gene is a section of DNA that codes for a protein. The information that DNA carries is the code for proteins. Okay, so that's what we're going to get into next week. How does this carry that code and how does it get translated into proteins? But this week, we're just going to talk about what is this thing that I'm holding in my hand. A model of it, at least. So let's jump in and let's talk about the DNA structure. So DNA, deoxyribonucleic acid, is a nucleic acid. It's a type of biological molecule. There's four basic types of biological molecules, include like carbohydrates, proteins, lipids, and nucleic acids. Nucleic acids is one of those. And nucleic acid's main job in biology is to carry and store genetic information. And genes create proteins. So it's the information to create all the proteins which do all the work of life. So, nucleic acids are this type of molecule. And there's two types. We're going to talk about DNA today, and then we'll talk about RNA next week. They're very closely related in structures, a couple little differences between the two. So, it is a nucleic acid, deoxyribonucleic acid. And nucleic acids are made up of these tiny little units. They call them structural units, or just units, right? So, this is a nucleic acid, the whole thing, DNA, deoxyribonucleic acid. But what is it built of? It's built of little structural units, and those structural units are called nucleotides, okay? Nucleotides. Nucleotides, this is a model of one here, this little thing, clicks together, and these then stack up again and again and again to make up the whole nucleic acid. The analogy there would be a nucleotide is like a brick, and a nucleic acid is like the brick wall, okay? So this is the part, and this is the whole. Nucleotides, okay? And nucleotides themselves can be broken up into three little parts. So a nucleotide has three main parts. Nucleotide is made up of a phosphate, a sugar, and then a base. You'll see nucleotides written different ways. So there's your phosphate. It'll say phosphate group sometimes because it's a phosphate with oxygens around it. You don't need to know that. Just know phosphate. And then the sugar. It can also be called a 5-carbon sugar or a deoxyribose sugar. I just say phosphate and then sugar right there. And then lastly, the base, right? Right there. The nitrogen base in the middle. And so you see that here on this model as well, on the screen. You see the phosphate and the sugar phosphate, sugar on the sides. And then in the middle, they have different letters for these, and we'll discuss those in just a minute for the bases, because bases have different names, but the nitrogen base in the middle, or a base containing nitrogen. Okay, so once again, nucleic acid, made up of these little parts here. You break them out, nucleotides, and then a nucleotide is made up of a phosphate, a sugar, and a base. Phosphate, sugar, base, phosphate, sugar, base. Say it again. Or don't. Or don't. Okay. So here we go. And this drawing shows it as well. You've got the big double helix, double-stranded nucleic acid. And then when you look at it, you've got the phosphate group, pentose sugar. It's a 5-carbon sugar. And then in the middle, the bases, right? And these link up again and again and again and again and again. This nucleotide makes up the whole nucleic acid. Some vocab, and it's layered, but you got this, okay? And there are a number of different ways to draw nucleotides. So when you look them up on the internet, I just grabbed a bunch of examples. So here, phosphate, sugar, base. Again, a nucleotide is 3 parts. And then another drawing of the same thing, and a drawing of the same thing. And another drawing of the same thing, right? It's that phosphate, sugar, base, again and again and again, making up the nucleotide, makes up the nucleic acid. I know I'm saying the same thing over and over again, but for some people, this is kind of hard to struggle. They struggle with this because of sort of the layering of the vocab, right? So the smallest parts would be the phosphates, the sugars, and the bases. That would make up the nucleotide, be the medium part, and then the nucleotides make up the largest part, the nucleic acid. There you go. I explained it again. Ha! All right. Now, if you'll look at this diagram here, on the outside you see the phosphates and sugars. Right here, right? In the outside rungs, you see phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And in the middle, you see the bases. What is the one part of this molecule that changes? On this DNA, as you go up and down it, what is the one part that changes and varies and is not consistent in the pattern that you see? I'm not trying to give too much away here, but if you just look at this right here, phosphate, sugar, phosphate, sugar, phosphate, sugar on the outside. And then up the middle, you see the bases, but there's something funky there. The only difference between each of these nucleotides is the type of base that is on it. So with the nucleic acid, right, nucleotide, phosphate, sugar, but notice the colors here, the bases are different. So there are different bases. There are 4 different bases. And you see those circled here. These are different here. So that is the part that varies and changes as you move up and down the DNA molecule, is the type of base that is located there. And there are 4 bases. Okay, these commonly get referred to as just A, T, C, and G, but they do have names, and I want you to learn them. So here we go. So the base with an A is adenine or adenine. The base with a T is thymine. Say it with me. Thymine. Kind of cool. All right. The base with the G is guanine. And then the base with the C is cytosine. Couple fun facts here as you write these down. Write down the not only the A, T, C, and G, but then the actual full name. Fun facts about these, they end in INE, and in chemistry, when you hear something that ends in an INE, an ine, it is a nitrogen-containing molecule, or an ene. So all of these contain nitrogen. You can tell that by the end of their name there. Another fun fact, guanine. You may have heard of a substance called guano, and guano is, let's just look it up. Guano. Yes, that is it. Guano is poop, bird poop or bat poop, and it is exceptionally high in nitrogen because it is exceptionally high in guanine as a byproduct, as a waste product that gets given off by metabolic processes, and it's used as a fertilizer around the world. It's gathered up and used as a fertilizer, and also has been used in explosives and things like that because it does contain nitrogen. So old school, like road building and stuff, they would use bat guano. In other countries, they still use that as well. Fun fact. All right, so those are your 4 bases there that you have. Again, adenine, thymine, guanine, and cytosine. Yeah, let's keep this party rocking. Okay, you do not have to know like the pyramines versus the purines and the different families or groups that these fall into. It's basically if they're single ringed or double ringed, and no need to worry about that for now. You will, however, notice a certain pattern here, and it's called the base pairing rule. So if you look up and down the molecule, right, of DNA, you'll see a few things consistently, and this is what makes this molecule so special, is that there are rules with it. And those apply across all living species here on Earth. We all adhere to the same rules. We all actually speak the same biological chemical language, which is cool and shows relation as well. If you look up and down the molecule, you'll see a few things, right? You'll see that phosphate, sugar on the outside, up and down each side. You'll see the bases in the middle, okay? And you'll see that the bases change and the pattern of the bases varies. So you may get an A, G, T, C, you may get an A, A, C, T as you move up and down it. But you'll see one more pattern in here, one more sort of rule that has to do with the bases, and if you can imply base pairing rule, it has to do with how the bases join in the middle. So take a look at this again, either one of these images, and see if you see a certain pattern in how the bases bond together, or which bases bond together. Ding, 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 ding. That's right. Oh, I should ring my big bell. The base pairing rule states what? Yes, it states that A will always bind or bond with T. Notice AT, 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 or TA, you know, genetics, it can be dyslexic. And G will always bind to C. Base pairing rule. A always pairs with T, G always pairs with C, right? Or you could reverse that. So as you move up and down this molecule, right, there's consistency in every single species here on Earth. We adhere to this structure and we adhere to this pattern and to how it gets read out and coded. So that's what allows us to manipulate genes across species, or actually move genes across species and have them still produce the same products. So phosphate sugar on the outside of this nucleotide here, and then in the middle, boop, you got the base, and the bases vary, but even if the bases vary, they pair in a consistent pattern. Wherever you have a T, you would have an A on the other side. If you had an A on one side, you'd have a T on the other side. If you had a C here, you'd have a G there. If you had a G there, you'd have a C there, T here, A, G, C, right? They always pair with each other. Another way to think of it, remember it, is that the curvy ones always pair together, the G with the C, and then the real angular ones, the A and the T, always pair together as well. That's kind of a helpful hint. Study tip there. Okay, so DNA is made of two strands of nucleotides, right? So the nucleotides form and pair in the middle here. So this nucleotide, this phosphate's going to bond with this sugar here, and then this one is going to bond over here, phosphate sugar, and then the bases will pair in the middle, and they pair up in a kind of a ladder structure, and then they twist. It's called a two-stranded spiral or a double helix. Yay! And again, the sugar and phosphate on the outside, and the bases, paired bases, connecting on the inside there. Now, the reason why DNA twists, this is pretty cool actually. The reason why it twists is, let me come up with a good way of showing this. Yeah, there you go. So if it's just a straight ladder, okay, it is less strong structurally, right? It can get ripped and torn. But if you twist something, it actually strengthens it. Do it with a piece of paper, and you'll see what I'm talking about there. And the other thing it does is it shortens it. So you're able to store more DNA inside the nucleus of a cell, and it compresses and compacts it. So the twisting shortens it, just like a spiral staircase would. But it also puts the phosphate sugars on the outside and puts the bases in the middle and protects them, because that is where the information is carried, and that's what we'll talk about next week. All right, so that's it for the DNA structure notes this week, except for honors. You guys stick around. Everybody else, go ahead and work on the DNA notes assignment 2 for this one, structure. There's just one sheet there, and I did include some images from the slideshow to help you out in answering those questions there. And now, honors, let's stick around and go to the next slide, shall we? Boom. DNA replication. Okay, so hang on just one second with this. I want to see if I can do a trick. I don't think I can. Let's just keep going. Okay, so DNA replication. One of the things about the DNA molecule is that it can make exact copies of itself, and it does again and again and again, okay? You are the result of that. Every single person watching this video, every single living thing here on Earth, be it this cucumber or this, well, no longer living piranha, or me, started out as a single cell, right? We started out as a single cell, and that cell has divided, and then those cells have divided, and then those cells have divided and divided and divided, and we've grown. All living things grow, right? Living things grow and reproduce. So here we are, having this conversation with a cucumber, or as the English like to call it, a gherkin. Okay, started out as a single cell, and now I'm 37 trillion cells, and this cucumber slash gherkin is, I don't know how many, probably millions of cells large itself. And the information has been passed down amazingly well, right? Here I am, I'm still functioning, right? So the code stays intact, and that code gets written again and again and again and again and again. How does it make exact copies of itself? How does that one cell make 2 cells, make 4 cells, make 16, make 37 trillion, and keep that information intact each time? How does it pass along that code? So there's a couple things that we've learned today about DNA that allow it to do that. One is that complementary base pairing rule, right? It's that the DNA molecule A pairs with T and C pairs with G each time as you move up and down it, right? And the other is that it is 2-stranded. It's a double helix. So you've got 2 copies of that code. So here's one clue for this right away. First, what you have to do is you have to break it down the middle, snap, right? You're still keeping it intact, but you're starting to separate this molecule. So you're going to break it down the middle. Let's see if I can do that here. Boop. And the bonds get unzipped, basically, down the middle, and it separates out now. The next step is to then start adding in new bases, new nucleotides on each side. So if I separate out here, and I have a T here and an A here, and I separate them over here, I'm going to hide this one. I have a T here, I'm going to pair an A to here. If I had an A here, I'm going to end up with the T here. So now I have a TA, and I have a TA here. Now you're starting to see it. I have 2 copies. You do this all the way down the entire thing. First step, right? You have your double helix number 1. And then 2, you unwind and separate out. 3, add in new nucleotides. Add in new nucleotides and pair each base accordingly to the base pairing rule. And then step 4, right? Rewind and then recoil along that phosphate sugar base on the outside, phosphate sugar on the outside. And the way that it does this is pretty cool and interesting. There are certain proteins, because proteins do the work in biology, that help in this process. The first one is called helicase, and that protein is the protein that goes down and actually unzips, right? It unhelicases the molecule. And so yeah, it basically goes and breaks all these. These are hydrogen bonds, so they're not very strong bonds, joining the bases in here, and it just snaps those apart and kind of unzips it. The next is DNA polymerase, and DNA polymerase is the protein that goes along and reads and then matches new nucleotides to the different to the bases along each strand accordingly. And then ligase comes back along, and I believe it actually proofreads, if I'm correct, or it rebonds it as it reconnects. Okay, so there you go. Here is a larger example of it here with the helicase going through and unzipping, the DNA polymerase going along and then matching up those new nucleotides along to the respective bases. So a real simplified drawing would be this. So here's your parent strand of DNA, your original one here. And then what you do is you break it in half using the helicase, so you unwind and unzip it, and then you match up on each side, right, to its respective bases, new nucleotides along there. And then what you end up with is 2 identical, what they call daughter strands of DNA. This happened in your original first cell, and this has been happening to every cell in you that has replicated ever since you were born. And not only are we made of 37 trillion cells, so this has happened 37 trillion times at least, this has happened way more than that because your cells die and then new ones are made from old ones and on and on and on and on. Now, this isn't always done perfectly. Sometimes there are errors in the reading and matching of this code. And those errors can be nothing. They can be a wasted attempt of that cell, and so it has to start over again. But sometimes they can be passed along. Right? Those errors are mutations when the code is read wrong. And so those can cause things like aging, and they can also cause diseases, things like that, like cancer even. So we can hit on that one later. Okay, I put a couple links up here for you guys. The Learn Genetics has some great interactives on there, so check that out. There's a whole bunch, and it's all DNA related stuff, and especially after next week, you'll really be able to understand, but if, you know, just after this week, you'll be able to grasp and understand and appreciate so much more of what's going on here. And then I put an article on DNA replication and recombination. And if you're curious about kind of how better to answer this question that I posed to you, you can read that. Although the last question is 5 points, I want you to just explain as best you can in a paragraph, right, this question here. How does the double helix structure of DNA, along with the base pairing rule, allow for it to make exact copies of itself? So use your vocab and just describe what we just talked about right here. All right. Thanks for sticking around and enjoy.